For pretty much all of human history, the code of life was something we were just stuck with. We could read it, sure, we could study it, but we couldn't change it. So, what if we could suddenly pick up the pen and rewrite the story ourselves?
Just think about that for a second. So many of the diseases that have plagued us for centuries, things like sickle cell anemia, cystic fibrosis, Huntington's, they're all caused by these tiny, devastating typos in our DNA. We've always just treated the symptoms, right? Because the root cause was untouchable, locked away deep inside our cells. Well, that's not true anymore.
Our genome is basically the most complex instruction manual ever written. But, nature isn't a perfect editor. Mistakes happen, and those tiny mistakes can have absolutely massive consequences. So the big challenge for medicine has always been, how do you fix just one tiny error without messing up the entire blueprint? We needed something like a find and replace function for our very genes.
And the amazing thing is, the solution didn't come from some brand new futuristic invention. It actually came from a discovery, a bit of incredible scientific luck. It turns out the most powerful gene editing tool ever known wasn't created in a high-tech lab. It was found hiding inside some of the simplest organisms on Earth.
It's called CRISPR. Yeah, I know. It's a mouthful. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. But what it is pure genius. See, for billions of years, bacteria have been at war with viruses. And CRISPR, that's their immune system. It's like a genetic library of all the viruses that have ever attacked them. It lets the bacteria recognize and just shred the DNA of any virus that tries to invade again. It's like a genetic memory card.
And this timeline just shows you the incredible journey this technology took. It all started way back in 1987 as just a weird observation, some strange repeating DNA. For almost 20 years, it was just a puzzle. Then, boom, 2005, the aha moment. Scientists realized those repeats are like viral mugshots. By 2012, Emmanuel Charpentier and Jennifer Doudna figured out how to turn this natural defense into a tool we could control. Just 1 year after that, Feng Zhang got it working in human cells. And by 2020, it won the Nobel Prize. That's how fast this thing took off.
So, how exactly did scientists manage to take this ancient bacterial weapon and turn it into this ultra-precise biological tool? How'd they hack the system to make it work for us?
Well, they basically boiled it down to two essential parts. First, you've got an enzyme called Cas9. Think of this as the molecular scissors. It's the protein that does the actual cutting. And second, you have something called a guide RNA. This is the GPS. It's the programmable part that tells those scissors exactly where to go and what specific DNA sequence to cut.
Okay, so here's how it works, in 3 simple steps. First, scientists create that guide RNA to perfectly match the gene they want to change. Second, that guide leads the Cas9 scissors on a search through the entire genome until it finds its exact match. When it does, Cas9 snips the DNA, making a clean cut. And third, and this is the really clever part, the cell's own natural repair crew rushes in to fix the break. Scientists can then trick that repair system into using a new, healthy piece of DNA as the template, effectively rewriting the code. And that molecular scissors analogy is just perfect, because it really gets at the incredible precision we're talking about. This isn't some clumsy brute force method. It is a biological scalpel that can sift through the 3 billion letters of our genome to find one specific spot and make one single clean cut. It's just amazing.
And this is why CRISPR caused such an absolute explosion in science. Before CRISPR, we had other tools, but they were incredibly expensive, really slow, and super complicated to use. CRISPR changed everything because it's cheap, it's fast, and it's easy to program. It basically put the power of gene editing into the hands of thousands of labs all over the world, practically overnight.
And with the tool this powerful and this accessible, you better believe the applications have been expanding at a dizzying pace. We are way past theory now. CRISPR is actively changing our world, from medicine to agriculture and, well, everything in between.
In medicine, the potential is just staggering. We're already seeing clinical trials where they're successfully treating sickle cell disease by editing a patient's own blood cells. Researchers are literally engineering our own immune cells, T cells, to turn them into superpowered cancer hunters. There's even work being done to edit our cells so they're completely resistant to viruses like HIV. It's a total shift from just treating symptoms to actually fixing the problem at its source.
And it's not just about human health. In agriculture, CRISPR is creating crops that can survive drought and fight off pests, which is a huge deal for food security.
In fact, in Japan, you can already buy a tomato that's been edited to have more of a healthy compound called GABA. And then there's the really wild stuff, like developing gene drives that could spread a trait through an entire mosquito population, maybe even wiping out malaria for good.
But with this kind of incredible power comes an enormous weight of responsibility. The ability to correct genetic errors naturally leads us to a much more difficult question. Should we also use it to improve the code? And this is where the science crashes right into a massive ethical debate.
This whole debate went from theoretical to shockingly real back in 2018. A scientist in China announced he had used CRISPR to edit the genes of human embryos, leading to the birth of twin girls. Now, his goal was to give them resistance to HIV, but the experiment sparked global outrage. It was condemned as reckless, unethical, and it proved that the technology was here, and we were not ready for it.
And that event threw two completely opposite worldviews into the spotlight. On one side, you have thinkers like Julian Savulescu. He basically argues that if we can use technology to give our kids a better, healthier life, we have a moral obligation to do it. But on the other side, you have bioconservatives like Michael Sandel. He warns that this constant drive for perfection is dangerous, that it makes us lose our appreciation for life as a gift, with all of its unplanned, messy, beautiful parts.
The philosopher Jürgen Habermas really put his finger on this fear. What he means here is that the second we start designing our children, we change everything. A child stops being someone we welcome into the world and becomes a product we've manufactured, and he feared that could completely undermine the very ideas of human freedom and equality.
And his concerns are part of this bigger cloud of risks. There's the terrifying idea of a new kind of eugenics, where we'd select for traits that society deems desirable. There's the real risk of creating a genetic divide, a world of the genetically enhanced rich and the naturals. And of course, there's that slippery slope. Where do you draw the line between fixing a terrible disease and just enhancing a normal human trait? These are some of the hardest questions we've ever had to face.
So, this brings us to the biggest implication of all. For the very first time in the history of life on this planet, a single species, us, now has the power to consciously and directly guide its own evolution.
And that's exactly what this captures. For 3.5 billion years, life was shaped by the slow, blind process of natural selection. We were just products of that process. But now we're becoming the architects. We're going from being characters in the story of life to its authors.
Look, the scientific question of if we can do this is over. The technology works. So the focus has to shift from the science lab to the public square. The really hard work ahead isn't scientific. It's about all of us deciding, as a global community, what the rules should be.
At the end of the day, we have to remember that CRISPR is just a tool. It's like fire or nuclear power. It isn't inherently good or evil. Its impact is going to be determined entirely by the choices we make, by the values we prioritize, and by the wisdom, or lack of wisdom, we bring to the table. And that's the ultimate question CRISPR leaves us with. We are standing at a monumental crossroads with the power to edit our own source code. So, what kind of future are we going to write for ourselves? What story do we want our genes to tell?